and try to finish on time. We have a lot of information to cover, and I'll start off talking about the human genetic gene discovery part of the project, and then I'll be joined by Eric Liao, who will talk about the zebrafish models briefly at the end. So without further ado, most of you remember that the basic organizational structure of our SCOPE project consisted of three aims. They were, first of all, to find patients with bioinformatically tractable and developmentally interesting craniofacial disorders that were likely to be monogenic. Once they were ascertained, and I should thank our wonderful collaborators there, which is Eric Liao, of course, at Mass General, and David Sweetser, Catherine Noack, and Joan Stoller at Boston Children's Hospital, Pedro Sanchez at LA Children's, and then now at Cedars-Sinai, and then Ophir Klein and Hazel Perry at UCSF. With that, we've subjected these cases to whole exome and to a lesser extent whole genome sequencing, used our bioinformatic pipeline to find the genetic variants, and then in some cases validated those functionally in zebrafish, in other cases, and more efficiently where similar developmentally related phenotypes exist. We have made use of that human genetic axiom to establish causality. Our pipeline was discussed in a recent publication in Genomic Medicine, which I'll refer you to if you're interested in that further. Just by way of summary, I've shown this slide before. Many of you remember it. Everybody knows, of course, that these dominant pedigrees are obviously likely to be monogenic in nature, but the real developmental sweet spot for the cases that we're looking at are over here in the de novo dominant disorders, where we sequence the parents, the affected proband, usually just one affected family member per case, and the bioinformatic filters are set in such a way using inheritance mode specific callers that are very sensitive to just generate 0 to 3 mutations per case, and those have turned out to be remarkably tractable from this approach. There's some assumptions: monogenic inheritance, complete penetrance, and then whole exome. We're only looking at coding sequence. Here's our summary of project throughput to date. Some of the analysis are still underway. Our original goal was to try to solve 25 novel cases over 5 years. There were several misconceptions about that. I realized once we got underway. One was that a significant finding has been we have, quote, rediscovered phenotypic expansions of known syndromes that couldn't have been diagnosed clinically because of the imperfect state of dysmorphology. And then another smaller fraction turned out to be novel gene discoveries. So we evaluated 130 cases over 5 years. We accepted 112. Some were rejected because of incomplete family structure, or they weren't good cases for one reason or another. We've sequenced 45, and the basis for this large discrepancy is that about 38 of those cases that we accepted, we could not obtain full consent to do the studies. Another 14 or so, we were able to get consent, but can never get the blood samples for sequencing. However, of the 45 that we got, most being exomes, a few genomes, typically trios, that's why there's about 3 times the number of samples sequenced as cases analyzed. And then we solved, or provisionally solved, and I'll explain what I mean by that, about 28 of them, which is a remarkably high average for these complex cases, we think. And then we have about 14 models that have been made, largely targeted deletions using CRISPR. So here's a summary of the 28 cases. The ones that have the yellow asterisks on the left are ones we consider pretty conclusively or conclusively solved, either by nature of the fact that the phenotype was recapitulated in other individuals with the same mutation, or I should say a mutation in the same gene. Other ones are narrowed down to a single candidate gene, and that's usually what we mean by provisionally solved. We have a high index of suspicion that's correct, but the laborious work of doing functional reductionist studies in animal models or finding additional human cases is still underway. And I'll talk about some of these in the next 5 or 10 minutes. I've talked about a couple that turned out to be genes already linked to unusual disorders, but which were known. One of those is POLR1A, another is NFIX. Then I'll talk about some newer cases that we've looked at. One theme that's emerged is we start to find recurrent hits in gene family members. One example here would be the ubiquitin ligases HECD1 and HECD4. Those aren't really proven to definitive resolution, but we're pretty convinced those are meaningful targets. Another one are the mediator complex components, MED12 and MED13L. And so some of these turn out to be very sensitive key developmental regulators that interface with known signaling pathways, and that might be a theme that's emerging from these somewhat agnostic studies. All right, what kinds of results do we get? Really, the kind that we'd hoped is really about a third or less of the ones we saw. Those are where we find a new craniofacial disease gene. One of our favorites is piezo2. That's triggered a bunch of reductionist studies, which I won't talk about, but it's turned out to be fascinating. That's a mechanosensory ion channel, and likely explains some novel biology that we've seen. In other cases where we land on a gene that's a known craniofacial or other disease-causing gene, and so the reason it wasn't diagnosed clinically is because the phenotype deviates from the classic phenotype in certain ways by having other ancillary findings, and we call that a phenotype expansion. Another case is that the diagnosis couldn't have been made clinically, and one reason for that is simply because of the subjective nature of dysmorphology as a field. I think the work that Rich and Ophir are doing is very notable in that way, in that they're providing much more precise metrics of craniofacial parameters in some of these disorders, and we think that plus with sequencing could really help to transform dysmorphology into a much more systematic field. And then, of course, we have another fraction that we don't solve. So basically, the way to think about this is we have about a third of the ones we can solve are new craniofacial disease genes. Another third are phenotype expansions of known disease-causing genes. Another third basically are just because of the variable expressivity of known phenotypes, and then there's some that we don't solve. The clinical ramifications I've learned, and I've said before, are really quite significant in that when you can provide a family with a molecular diagnosis, it's only good so far. The family is remarkably grateful to know that they have a known disease entity. They can tap into support groups. There are downstream surveillance that can be done medically for problems that are yet to ensue but are known because of the natural history of that particular class of mutations in that gene. And it really has been quite remarkable to see the beneficial effects. All right, two quick recaps of cases that I presented before just to tell you what we're dealing with here. This is a case of Pedro Sanchez, profound craniofacial dysmorphosis. Which one? Not clear. We sequenced the trio. Basically, we land on a gene of basically the RNA polymerase 1 subunit POLR1A, which we then learned was associated with an acrofacial dysostosis Cincinnati type. So even a world-class dysmorphologist like Pedro would not have likely been able to make this diagnosis in the clinic. And in fact, that's a missense change. There are other missense changes in the literature, and there's a zebrafish model that already exists. This black arrow down here at the bottom is Meckel's cartilage. You can see in this zebrafish mutant made by Paul Traynor, it's basically missing, and that's a pretty conclusive proof. Another case is this sagittal craniosynostosis. This child was sequenced as a trio. We found a de novo splice mutation in exon 6. This is known as Marshall-Smith syndrome. NFI-X is the gene. What was interesting here is this child had a glioma in the brain. It turns out that NFI-X is repressive for glial differentiation, and that likely explains this. So this tumor is now being followed by active surveillance. We've gone on to look in glioblastoma multiforme to see if there's a representation of NFI-X loss of function mutations. That work is still underway. So, okay, a few more cases then that'll illustrate different features of what I've been talking about. I'll jump right in. This is a case of Joan Stollers at Boston Children's Hospital with lobar holoprosencephaly. Just to remind you, holoprosencephaly has about 3 or 4 clinical flavors, depending on the degree of fusion of the brain. So in the case where the brain is fused partly, we have what's called a semilobar holoprosencephaly, illustrated in B here. The extreme case, of course, is when there's alobar HPE. The case we're dealing with here is sort of intermediate between semi-lobar and lobar, and you can see that the brain is really not separated into hemispheres in either of these two cases. In the lobar form, it tends to be partly separated with fusion at the midline. So in this case, we sequenced this trio. This is a more typical kind of family where there's multiple complications. One is that it was known from clinical sequencing that there's a GLE2 variant segregating in this family. GLE2 is in the sonic pathway, would seem to be a potential candidate for explaining HPE, except that basically these individual family members did not have HPE. And in addition, it's been shown by work by Max Monkey and others that these particular GLE2 variants don't tend to cause or associate with HPE. So we sequenced the trio, sequenced the unaffected family member, and basically landed on these genes here, one of which is CARD8, and that's a de novo mutations. The de novos seem to have a very high chance of being significant. This is a fairly dramatic change to a phenylalanine from a leucine. And when we looked this gene up, we found out it's known to interact with the patched receptor as a accessory factor that facilitates the transmission of a caspase-9 patch-mediated cell death signal. And so the hypothesis there is that this mutation, L236F, has another name as well, depending on the isoform you're looking at. The hypothesis is that that's a gain of function to trigger patch-mediated apoptosis and explain the midface hypoplasia and clefting in this family. So that's an example where we can turn to the zebrafish model to inject a RNA that is this presumptive gain of function allele. Instead of making a knockout, which Eric will explain takes a long time, even in zebrafish, these experiments are underway for some of the ones that are not simple loss of function, or at least the hypothesis is not loss of function. All right, here's another case. This one came from Ophir and Hazel at UCSF. Cleft lip and palate, developmental delay. It was thought the child had autism, but that was never really documented. There was hearing loss, constipation, and obesity. Normal other evaluations, genetic testing was normal, as is so often the case in these types of cases. And this is the differential down at the bottom. So we sequenced the trio and basically found that there were two interesting variants. One in the gene, or I should say two, in the gene called HNRNPK. That's a nuclear ribonucleoprotein component that's involved in ribosome biogenesis. But you'll see that these are basically splice site, extended splice site changes. We've learned that some of these can be significant. These are not in the canonical core splice donor acceptor. They're usually several base pairs, in this case 7 base pairs upstream of a splice donor, or 3 base pairs downstream. Actually, I have that backwards. 7 bases downstream of a donor, or 3 bases upstream of an acceptor. You can't really be sure without testing them. But in the meantime, we found another variant in this gene MED12 that is a single base pair insertion of a G. Instead of an A, there's an AG dinucleotide that causes a frame shift. Now, the analyst analyzing this case looked up in NOMAD, the genome annotation aggregation database, and found that there is a common polymorphism that inserts, actually, if you count this, you'll see there's 13 nucleotides here in place of the A. You got 13 nucleotides, so there's a gain of 12. So that's not a frame shift, as it turns out. And what we went on to realize that this falls in a portion of the protein where there's a glutamine, polyglutamine tract. And these bases here that are being inserted just add another run of additional 4 glutamines. So that variant is probably of inconsequence because, as in the zebrafish sequence at the bottom, you can have extra glutamines in this exact region, presumably without a functional effect. However, if you cause a single base pair change here, you'll get a frame shift, and you would truncate then the C-terminus of this mediator, which is, of course, an essential component of RNA-mediated polymerase activity complex. So the way this works is MED12 assembles with Pol, and then in the case of these mutations, there is a disassembly of this complex, loss of repression, and you get upregulation of, in this case, GLI target genes. So this is a Masonic pathway. It's one of many mediator components. And so the idea is that this truncation mutation causing haploinsufficiency, we have a PLI score to help compute the significance of that. That looks at the sensitivity of losing one copy. And of course, MED12 is on the X chromosome. So that's a very high likelihood mutation. There's also a syndrome.
Okay, you're at 5 minutes, just to let you know.
All right, and I will finish up on that note, except to say that we found additional MED mutations, this one being one, another one from Ophir. I'll just jump to the end of that, showing that there's a single base pair deletion here causing a frame shift in MED13. So these two proteins assemble in the same complex, and we think that's an important general feature of the types of genes we're getting out of these analyses, where we're finding multiple genes landing in the same area. There's also evidence for this to support this from the literature and also from the zebrafish. So I'm going to turn over to Eric here in the last few minutes to talk about the zebrafish studies. In this particular case, I'll just say that you can make a morpholino knockdown more easily than a mutant, and you can rescue that. So here's the morpholino in the middle here. You get this tail curving phenotype. You inject in a mRNA, and you can fully rescue it. So this will be a good opportunity to inject a truncated MED13 or MED13L homolog 13B and see if it's really functional or not. So those are the types of experiments. Now, what Eric's going to talk about are simply some of the loss of function strategies in zebrafish. So Eric, you want to take it over from here?
Hi, can everyone hear me?
Yep.
Yes.
Okay, good. Technology, you never know. So first, I just want to start out by thanking the group and Dick for the opportunity to work on this project. It's been incredible for a clinician scientist to be able to take care of the patient, but then also work on the sequencing and then try to figure out the mechanism in animal models. So as Dick alluded to, there are two potential genetic scenarios. There's the gain of function, where it's easier for us to model, where we could express the mutant protein in the zebrafish and can see a phenotype right away in the F0, versus the loss of function, where we have to go through a breeding scheme, usually, as you see in this diagram, to F3, where each generation takes a minimum of 3 to 4 months. So even though the zebrafish give us greater numbers, we have to still wait about 1.5 years to see the mutant phenotype. The other issue with zebrafish, as many of you may know, that whereas in mouse or in iPS cells, we're doing homologous recombination, in the zebrafish, we are really doing targeted mutagenesis with Cas9. So we have a trouble modeling the exact gene variant, if that's what's going on. More commonly, we're creating a mutation that we will then screen for the loss of function phenotype. Next slide. If we keep going to next slide, Chris. So I wanted to then use these two cases to highlight some of the things we learned in working up these clinical cases. That, for example, FB44 and FB142, these are both cases of rare, complex oblique facial clefts. On the left, we have a whole pedigree of 4 patients out of 14. So we're able to sequence not only the parents who turned out to be heterozygotes, but also the unaffected siblings. In contrast, a more typical case is we see a kid in clinic, and he's a de novo case so we have parental DNA in trio. And this really highlights the ability for us to get the best genetics data from sequencing that really can help us downstream. Next slide. So that in the case of FB44, these poor patients have severe bilateral facial cleft. They have associated eye phenotypes where they had anophthalmia or coloboma. In this case, we defined a gene variant to be a missense mutation. A missense mutation on the list of genes that we may not have been able to pick up if it wasn't for the fact that we have also the unaffected. so that even though it's a missense for the mutation in the homeodomain, it's quite obvious that likely it will be pathogenic. Next slide. So here, another approach we've taken in the lab is to have complementary models so that we can use iPS technology to derive neural crest cells from the patients and examine the neural crest cells to see if we see a phenotype in culture. And from those observations and characterizations of the neural crest, then form hypotheses that we can test in the zebrafish. And for both of these cases, you'll see that it's been very successful for us. So here we not only were able to derive the neural crest cells from the heterozygote parents and the unaffected children and the affected, here we also were able to make the CRISPR mutants in this gene ALX1. Next slide.
We're at time.
Okay. So in brief, then we were able to model the phenotype in the zebrafish so that we can ablate the frontonasal process, which is regulated by this ALX1 gene. And we also show from the neural crest cell studies that these cells actually remain in the progenitor state. Next slide. And we showed that they have a migration defect. We can see the same migration defect in the lineage tracing of the zebrafish animal. We found that these neural crest cells is a subset that actually is a common progenitor for not only the midfacial tissues, but also the eye, explaining the eye phenotype in the patients. Next slide. In contrast, in this other case, which is also complex facial cleft that involves the orbit, but because it's a de novo, next slide, it's less clear which gene it is. There's a number of candidate genes that are missense or frame shift mutations, and the likely candidate at working up now is this gene TDGF1, where now we're also doing functional tests both in iPS model and also the zebrafish. Next slide. So in summary, while whole exome sequencing is a powerful tool for diagnosing unknown craniofacial dysmorphoses, it should be the standard of care because, as Dick mentioned, we're quite successful in solving over half of our cases. We're still challenged by variable expressivity, which is pretty common in craniofacial disorders. There clearly are modifier genes and epigenetics and environmental factors. That a new craniofacial disease gene discovery is much more difficult and time-consuming than simply sequencing because of the use of variabilities of phenotype, and also when you get down to the weeds of making the animal models, it's actually still quite time-consuming. The importance of the trio for whole exome sequencing, obviously the pedigree is much better. There are variants and paralogs of known disease genes, and these could be good candidate genes for disorders or related phenotypes. And the downstream functional analyses still need to be customized for each gene, for each case. And it's been an incredible learning lesson for all of us involved in this project. Thank you.
Okay, great. Thank you. So we have a question from Timothy Cox from the chat. How many genes had variants in more than one family phenotypic presentation? Or are these primarily deemed to be pathogenic because they are de novo variants? Do all variants meet the current ACMG criteria for pathogenicity? And are the variance and pathogenicity scores available on FaceBase or elsewhere?
Yeah, so those are all good questions, Tim. Maybe we can try to take them in order. I guess the first question was how many have corroborating mutations in second families? I guess of the 28 that are solved or provisionally solved, obviously only the ones that are we consider definitively solved. I would say about 2/3 to most of those have second hits. In the cases where they don't, the argument is based on an existing mouse model or a zebrafish model, or in one or two cases, the biochemistry we think is sufficiently compelling from what's in the literature to make it an odds-on favorite. You also asked, I guess, about pathogenicity scores. That was the last question, which I remember. We're using a variety of tools for that. In general, and I guess you're meaning things like polyphen and SIFT, those have not been that helpful to us, even though polyphen was developed by the same person, Shamil Sanayev, who developed our computational pipeline. We're using now an integrated approach that looks at all the different types of data. First of all, representation of these variants in NOMAD has been very helpful. What's known about the biochemistry and biology, that's very helpful. Structural considerations, we've integrated that when a structure exists for the protein. That's been helpful. And basically, it's a informal weighting of those different factors that we're using. Whether this might ultimately evolve to an artificial intelligence type of model or something like that, I'm not sure. But that would be the idea. And maybe you can just repeat the two other intermediate questions, Chris.
Are these primarily deemed to be pathogenic because they are de novo variants? Do all variants meet the current ACMG criteria for pathogenicity?
I would say the answer to the de novo question is only probably about 2/3 of them are de novo, but that's not the reason we're invoking them as causal, although that's usually very good evidence, as I explained. As far as the ACMG criteria for pathogenicity, in general, they do meet that criteria. Some of them would be variants of unknown significance, though. Okay.
Okay, great.